Hello guys, so I'm going to give you some information about today's topic, why mitosis is so important, why it's required for growth, asexual reproduction, tissue repair, and embryonic development. Growth, multicellular organisms increase their size by increasing their number of cells. Asexual reproduction, certain eukaryotic organisms may reproduce asexually by mitosis. Tissue repair, damaged tissue can recover by replacing dead or damaged cells. And embryonic development, a fertilized egg zygote will undergo mitosis and any differentiation in order to develop into an embryo. Then we have that this is the cell cycle. What is the cell cycle? It's a series of events through which cell passes to divide and create two identical daughters. So we're going to study all this cell cycle, about all this cell cycle in the following pages, okay? But what do we need to know first? We need to know something about chromosomes, and we need to understand that every eukaryote has genes on chromosomes. Storage. What is a chromosome? It's a storage unit in the nucleus. As you can see, we have two chromatids, and every single chromatid has DNA that is supercoiled in this section, okay? Each chromosome has a partner, one from each parent. So one comes from your mother, one comes from your dad. Both copies are required for the cell to function. Different species have different chromosome numbers. In the case of humans, we have 23 pairs and 23 diploid number because we have two pairs, 46. In frogs, the number of chromosomes is 13 pairs, corn, 10 pairs, dogs, 39 pairs. And we need to understand what is a gamete. Gametes are sex cells, sperm and eggs. They are haploid, so just one end. They have a half set, as they will pair with the other half in fertilization. So 23 from one section, 23 from the other, male and female, okay? A normal somatic body cell has two sets of chromosomes, one set from the mom and another cell from the dad, in any kind of living organism, okay? When we talk about diploid, that means 2N, two set of chromosomes. Just before a prophase is the first phase of mitosis, the cell has two copies of every chromosome, okay? Has two copies of every chromosome, as you can see. They are called sister chromatids, these two, and they have centromeres. Centromeres hold sister chromatids together during prophase and metaphase. Following mitosis, each new nucleus is diploid. As you can see, they have the same genetic information. In mitosis, we're going to have like the cell, one cell that is that one is the pre-existing cell, and is going as a product. We're going to have two cells with the same genetic information. The first phase is interphase, consists of the parts of the cell cycle that don't involve cell division. It's like the preparation of cells. Okay, what happened in this preparation? Increase the volume of the cytoplasm, organelles produced, proteins synthesized. Then we have another phase. In interphase, we have three phases, G1, S, and G2. In S synthesis, DNA is replicated. Remember that we are going to have doubles. The product is two cells, so we must have cytoplasm and DNA in a double amount. And the G2, increase the volume of the cytoplasm, organelles are produced, and proteins are synthesized. So the cell is ready for cell division. In interphase, these are the normal functions, okay? Metabolic reactions, when respiration occurs to produce ATP. Protein synthesis, proteins and enzymes are necessary to allow cell growth. Organelles number are increased to first support the enlarged cell, and DNA is replicated to ensure a second copy is available to enable mitosis. And then we need to understand, okay? Mitosis is the division of the two nucleus into two genetically identical daughter nuclei. So what do we have? This is a chromosome. The size of the chromosome varies from 0.2 to 20 micrometers. This central part is called centromere. It's the part of a chromosome that links sister chromatids. As you can see, this is a sister chromatids. This is the DNA that is supercoiled. So imagine that you have your cell, Every single cell of your body has the same genetic information. In the nucleus, these chromosomes are like dispersed, and every single chromosome contains the DNA and the genes. For example, this section could be a gene that, I don't know, gives the color of the eye. Another section forms a protein. So depending on the genes that we have in the chromosomes, we are going to know the function of those genes, okay? Then we have these another centrioles. They organize spindle microtubules, as we studied in the organelles. A spindle, these are the spindle microtubules. They also refer to as spindle fibers, okay? In animal cells, two centrioles are held by a protein mass referred to as a centrosome. So we have the first phase, that is prophase. What happens in prophase? The centrosomes, as you can see, these structures, move to opposite poles of the cell, and spindle fibers begin to form between them. So these are the centrosomes, and these spindle fibers begin to form. As you can see, the other one has to move to the other pole. What else happens in prophase? DNA supercoils, chromatin condenses and becomes sister chromatids, which are visible under a light microscope. So they're going to be separated, okay? The nuclear membrane is broken down and disappears. That happens in the prophase. This is how can you see under the microscope this phase. Then we have the second phase that is called metaphase. This is how can you see in the microscope. So what happened with the spindle fibers from each of the two centromes attached to the centromere of each pair of sister chromatids, as you can see. So they are going to extend and they're going to attach to the two chromatids of these chromosomes. The same happened with the other one. In anaphase, continued contraction of the microtubule, spindle fibers cause the separation of the sister chromatids. They are going to be separated. These sister chromatids, like the arms of the chromosomes, are going to be separated. The chromatids are now referred to as chromosomes. Chromosomes move to the opposite poles of the cell. See? So they're in the middle. You can see that in the middle. You can see the chromosomes moving. In telophase, chromosomes arrive to the poles. Okay? Microtubules, spindle fibers disappear because they already accomplished their mission. New nuclear membranes reform around each set of chromosomes. See? So we have the two sets of chromosomes. They are going to have a new nuclear membrane. The chromosomes uncoil. They condense to the chromatin and are no longer visible under the light microscope. So what are you going to see? The spindle fibers, they are in the opposite sides. And these are the phases of mitosis, okay? Remember that now cytokinesis begins. What is cytokinesis? It's the division of the cell, the division of the cytoplasm in two different cells. As you can see, we're going to have two of the same cells. They have the same Golgi apparatus, reticulus, mitochondria, lysosomes, and as you can see. So remember, prophase, metaphase, anaphase, and telophase. People meet and talk. Remember that if you want to remember the order of these stages.